Good morning. It's March 11th, and this is your daily brief in science. Here's everything you need to know. A groundbreaking development in genetic research is on the horizon with the introduction of PhyloFrame, an AI tool designed to combat ancestral bias. Researchers at the University of Florida, led by Dr. Kylie Graham, have created this innovative tool to enhance precision medicine for diverse populations, addressing a significant gap in genetic data representation. Currently, a staggering 97% of sequenced genetic samples come from individuals of European ancestry. This bias raises concerns about the accuracy of genetic research and its applicability to underrepresented populations. The effectiveness of PhyloFrame was highlighted in a study published on March 10, 2025, in Nature Communications, backed by the National Institutes of Health. PhyloFrame sets itself apart by maintaining prediction accuracy across various populations, overcoming limitations of existing models that primarily utilize data from European ancestry. The researchers emphasize that incorporating diverse training data not only benefits underrepresented groups, but also enhances model performance for European populations, thereby preventing overfitting. To analyze the vast genomic data necessary for this research, the team utilized the University of Florida's Hypergator supercomputer, processing around 3 billion base pairs of DNA per individual. A key innovation introduced by PhyloFrame is the Enhanced Allele Frequency, or EAF, which identifies population-specific genetic variants in healthy tissue to improve disease predictions. The ultimate goal for PhyloFrame is to be implemented in clinical settings, facilitating personalized treatment plans based on genetic profiles while minimizing side effects. Dr. Grimes' interest in addressing ancestral bias was ignited by a conversation with a doctor concerned about the limited relevance of genetic studies for his diverse patient population. Despite the progress made, researchers stress the ongoing need for more diverse genetic databases to support equitable precision medicine and effective machine learning modeling. Pamela Björkman, a prominent biochemist at the California Institute of Technology, has been awarded the prestigious Wolf Prize for her groundbreaking research on treatments for HIV and coronaviruses. Her work has significantly advanced the understanding of the immune system's response to pathogens, particularly in T-cell recognition and immunization strategies. Since the onset of the COVID-19 pandemic, Björkman has concentrated her efforts on designing immunogens aimed at stimulating antibody responses against coronaviruses. The Wolf Fund recognized her contributions for providing new hope in the fight against infectious diseases through her innovative research. Her work is seen as a promising strategy for developing future vaccines that address major immunization challenges. Björkman is not the only recipient this year. The Wolf Prize in Architecture was awarded to Chinese architect Tiantian Xu for her transformative rural development projects in China. The Wolf Prize has been awarded annually for 47 years, celebrating outstanding achievements in the arts and sciences, with numerous laureates going on to receive Nobel Prizes. This year's other notable recipients include Jeffrey Dangle, Jonathan Jones, and Brian Staskowitz, who were recognized for their contributions in agriculture, physics, and chemistry. The prestigious history of the Wolf Prize is underscored by notable past laureates such as Stephen Hawking and Marc Chagall. The Wolf Fund praised Björkman's work as a new rational design strategy for future vaccines against major infectious diseases. In contrast, Xu's architectural projects have economically and socially revitalized villages through innovative designs, marking a shift from conventional urban development strategies. A recent study published in JAMA Network Open sheds light on a troubling connection between chronic pain and mental health. Approximately 40% of adults suffering from chronic pain also exhibit clinically significant symptoms of depression or anxiety, indicating a critical public health issue. Conducted by researchers at Johns Hopkins University, this systematic review analyzed data from 376 studies involving 347,468 individuals across 50 countries. The comprehensive nature of this study provides valuable insights into the mental health challenges faced by those with chronic pain. The findings reveal that individuals with fibromyalgia are particularly vulnerable, with over half showing signs of both depression and anxiety. Specifically, the study reported that depression rates were highest in fibromyalgia patients at 54.0%, while anxiety rates reached 55.5%. In contrast, those with arthritis conditions reported lower rates, with 29.1% experiencing depression and 17.5% experiencing anxiety. Younger adults and women were identified as being more likely to suffer from these mental health issues related to chronic pain, emphasizing the need for targeted interventions. Understanding the interplay between chronic pain and mental health is crucial, as individuals with mood disorders may be dismissed as having psychosomatic issues and could be excluded from clinical trials. The study underscores the necessity for routine mental health screenings in clinical settings. Dr. Rachel Aaron, who led the study, highlighted the importance of integrated treatments that address both chronic pain and mental health, as current therapies often treat these issues separately. While many individuals with chronic pain do not experience depression or anxiety, the significant rates among certain groups challenge the assumption that chronic pain is inherently linked to mental distress. This research highlights a pressing need for healthcare providers to routinely screen for mental health issues among patients suffering from chronic pain in order to better address this public health concern. A recent study from Weill Cornell Medicine has unveiled a pivotal discovery in the realm of blood cell regeneration. Researchers have identified FLY-1, a single molecular switch, as essential for activating blood stem cells to produce new blood cells, a breakthrough that could enhance the efficacy of bone marrow transplants and gene therapies. FLI-1 plays a crucial role by facilitating connections between blood stem cells and endothelial cells, driving these stem cells into a regenerative state and significantly boosting their expansion potential. This activation is particularly important for addressing challenges related to quiescent stem cells that have been impacted by chemotherapy or radiation. The study highlights that by activating stem cells with FLI-1, researchers observed improved expansion rates and successful engraftment in new hosts post-transplantation. This approach is especially relevant for patients who may have a reduced number of viable stem cells due to cancer treatments. Supported by multiple grants from the National Institutes of Health, this research involved collaboration with various organizations focused on advancing regenerative medicine. Looking ahead, the research team intends to further develop their modified mRNA approach for FLY-1, with plans to initiate human trials aimed at treating blood disorders. Blood stem cells, primarily found in bone marrow, remain inactive until mobilized, which is crucial for replenishing blood and immune cell populations. Dr. Shaheen Rafi, the senior author of the study, emphasized that this innovative method could significantly improve marrow transplants, particularly in cases where donor stem cells are limited. Through single-cell profiling, the study revealed FL1-1's critical function, noting that its absence keeps stem cells inactive and impedes their interaction with the vascular niche necessary for regeneration. Furthermore, the research found that higher levels of FL1-1 activity correlate with the greater regenerative potential of human umbilical cord-derived blood stem cells compared to those from adults. Published in Nature Immunology, these findings suggest that FL1-1 can greatly enhance the effectiveness of bone marrow transplants and gene therapies by aiding quiescent blood stem cells in transitioning to an activated state for rapid multiplication and maturation. A new study reveals a concerning link between microplastics and the health of our planet's ecosystems, specifically highlighting their detrimental effects on plant photosynthesis. This research indicates that microplastics, which stem from waste, are obstructing sunlight, damaging soils, and introducing toxic chemicals into the environment. As a result, terrestrial plant photosynthesis is reduced by approximately 12%, while marine algae photosynthesis drops by about 7%. The implications are alarming, with estimates suggesting that microplastics could lead to a loss of 4% to 14% in staple crops like wheat, rice, and maize. This could potentially add 400 million people to the already 700 million people affected by hunger as of 2022. Asia is projected to face the highest crop losses, ranging from 54 million to 177 million tons annually, significantly impacting wheat production in Europe and maize in the United States. In marine ecosystems, The study estimates a loss of fish and seafood between 1 million and 24 million tons per year, which constitutes about 7% of total seafood production. This loss could provide crucial protein for tens of millions of individuals. Researchers led by Prof. Huan Zhang from Nanjing University warn that the ongoing issue of plastic pollution could severely undermine global food security, especially as the world population is expected to reach 10 billion by 2058. Published in the journal Proceedings of the National Academy of Sciences, the study is based on over 3,000 observations from 157 studies, underscoring the urgent need for further research to validate these findings. While some scientists acknowledge the study's value, they emphasize the necessity for additional data to strengthen the predictions. Recently, global negotiations failed to produce a UN treaty to combat plastic pollution. But discussions are set to resume in August, where the critical need to address microplastics will be a focal point. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.